anumang hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng Chief Alternative for Personal Hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat, lilipad.
Recording in progress.
Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Maayos na bukas para sa Pilipinas. Maayos na bukas ating iparanas sa bayang Pilipinas. Ating abutin ang pangarap iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan.
Hello, good morning, everyone. Hi, welcome to the eighth installment of our Virology and Vaccine Institute of the Philippines, or the VIP program webinar series. I am Red Danganan from the Environment and Biotechnology Division of the DOST ITDI, and I will be your host for today's session. For this two-day lecture, we shall be learning a lot about the handling of novel and dangerous pathogens. This webinar is part of the VIP's Information and Education campaign on virology and other related topics. Also, this learning session is made possible by the collaborative efforts of the DOST Central Office, DOST ITDI, DOST PCHRD, DOST PICARD, and the DOST Balik Scientist program. Just a reminder, especially to our Zoom participants, kindly rename your Zoom account to the prescribed format, which is your affiliation underscore your full name. So, for example, I am from DOST ITDI. I will put DOST ITDI underscore Red Danganan. Don't also forget to mute your microphones to avoid interruptions during the presentation proper. Also, for the information of all our participants from Zoom, Facebook, and YouTube, you will get a copy of the certificate as long as you answer the evaluation form, which will be given at the end of the session tomorrow. Again, welcome to all our learners. We now have 162 participants on Zoom. Let me just check our YouTube and Facebook Live.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people, without compromising the safety of each and everyone.
recognizing the critical role of science and technology in economic development and progress. The Balik Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have an easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik Scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik scientist is to be not just a teacher or facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist program. Okay, being one, I really truly felt that Balik Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people. recognizing the critical role of science and technology in economic development and progress. The Balik Scientist Act, or Republic Act 11035, was signed into law last June 2018 by President Rodrigo Roa Duterte.
This is actually putting into law a program that has been started by the Department of Science and Technology almost 40 years ago. But we need some legal support so that we can implement it in a better way. The Balik scientists will have easier time in terms of coming here to the Philippines and rendering services.
Who is the Balik Scientist?
Experience and expertise of Filipinos who have made good practice of being scientists abroad, so that they can share whatever they have in terms of knowledge and wisdom to our own institutions, our own researchers.
My role and responsibility of a Balik Scientist is to be not just a teacher or facilitator, but also a pusher of innovations within the program.
Balik scientists are given support by the government for their stay in the country and are likewise provided with a wide array of benefits to ensure their maximum output.
The best incentives or privileges or benefits are having to be exposed with our farmers.
My greatest privilege would be doing collaborative work with fellow Filipinos.
The DOST, as the leading agency in charge of the Balik Scientist program, is tasked to facilitate the placement of the Balik Scientist among its priority areas from its sub-agencies. P-Card, PCHRD, P-Shared, Partnering with the DOST are the host institutions, private or public entities, providing the appropriate resources to the Balik Scientist in the completion of their research activities and other tasks.
I think the role of the institution is to give the space or the laboratory needed for the program or the project.
Working together, the DOST, the Balik Scientists, and the host institutions have proven the importance of collaboration and cooperation, critical of any nation's vision for success.
I am an advocate of Balik Scientist program. Okay, being one, I really truly felt that Balik Scientists would be able to help in uplifting the economic growth of the country.
My hopes is to really contribute to the space agency. It's very, very pragmatic, and we will need everybody's help, and also promulgate the STEM program here in the Philippines.
We really need to encourage other Balik scientists or other scientists abroad to give their time. They need to give back and help the country.
With the enactment of the Balik Scientist Act, the country is looking towards a stronger and more solid science and technology foundation, propelling the nation to further heights. Change has come indeed for science and technology. Science for change. Science for the people.
We are now living in a new normal. This COVID-19 pandemic marks as one of the global challenges experienced in this generation. It forces every sector of our society to innovate, in order to move forward. We, at the Industrial Technology Development Institute of the Department of Science and Technology, is trying every possible ways to continue our service to our people without compromising the safety of each and everyone.
Hello again, everyone, and sorry for the technical difficulties earlier. Okay, so now before we proceed to the meat of our session, let me first introduce to you our distinguished Balik Scientist and our resource speaker for this morning. Our speaker obtained her bachelor's in biology and a master's in microbiology at the University of the Philippines, Los Baños. She then pursued a UNESCO postgraduate course in microbiology at the University of Tokyo, then a doctorate degree in microbiology, major in animal virology at the University of Hawaii. She is a microbiologist and a virologist by training. She has over 15 years of experience in microbial systematics and culture collection related activities and over 20 years of shrimp and human diagnostics, including ISO accreditation and successful regulatory submission of in vitro diagnostic medical devices. During her doctorate studies at the University of Hawaii, she developed the first continuous cell line from shrimp. Upon her return to UPLB in 1997, she collaborated with the Bureau of Fisheries and Aquatic Resources to help alleviate the infectious disease problems in aquaculture. In the year 2000, she then pursued postdoctoral research at the University of Cambridge on viral discovery project and chlamydia diagnostics, where she also managed a BSL-3 facility. In 2002, their laboratory at the University of Cambridge established a spin-out company in the Bay Area in San Francisco, California, USA, called the Diagnostics for the Real World Limited. He has co-authored numerous articles published in refereed journals or as chapters in a book. She was also a reviewer for the Journal of Virological Methods in Aquaculture and an external reviewer for a PhD dissertation at James Cook University in Australia. She was also conferred several awards, fellowships, and certificates both in the Philippines and abroad. It is my pleasure to introduce a pride of our country, our very own Balik Scientist, Dr. Lourdes M. Nadala, to talk about handling of novel and dangerous pathogens. Please, let's give her a big virtual round of applause. Good morning, Dr. Nadala.
Good morning, Rinilyn, and thank you so much for that introduction.
You're welcome, ma'am. You go ahead.
Okay, so today's presentation is part of the first part of a two-part talk on handling of novel and dangerous pathogens. And this is a part of a series of the Balik Scientist seminar organized by ITDI and the Vaccine and Virology Institute of the Philippines. So some of you may have heard this already, but pardon me for repeating some of them, because this is part of safety is always an important factor in microbiology work, especially if you're handling pathogens and if you're handling an unknown microorganism for the first time, just like what happened to us last year when we were confronted with SARS-CoV-2. So I will have let me just share with you my screen. So I'll have this is just to show you this photograph of, on the left-hand side is an electron micrograph of a cell showing virus particles of SARS-CoV being released into outside of the cell into the environment or inside the human body. And on the right-hand side, shows you this is like a pictorial representation of SARS-CoV, and these orange structures look like a crown, so that's why it's called a coronavirus. And it has the envelope has an E protein, the S protein or the spike protein or the crown, the ribbon-like structures, and you have the matrix protein. Okay, so I just wanted to show you these pictures, which were actually from a CDC series of pictures. So there is some delay on. Let me just minimize my screen here. So this is just to give you an outline of my presentation. So to be able to understand why we have to observe or to have to practice safety practices and observe safety considerations, we would look at some historical background on lab-acquired infections or LAIs, and then we will move on to the rationale for biosafety considerations. And this would include things that we have to take into consideration for handling of novel microorganisms, especially if it's something new or novel and it's highly infectious pathogen. So there are some new emerging viruses, especially that have been discovered, and the latest of which is SARS-CoV-2. And what are the precautions that we can put in place in order to protect ourselves, the environment, and our co-workers? And some of them are already like part of good laboratory practice. And I would also talk a little bit about cataloging and biosecurity. So it is very important to take stock of what you have, especially if you have BSL level, BSL-3 or BSL-4 pathogens. So, and lastly for today, we'll also talk about transport and waste containment. So this is a very important topic that when you design a facility, for example, or a lab, do not forget waste containment considerations, because it is a very important part of any microbiology lab, especially if you're handling pathogens. And tomorrow I'll be talking about two specific examples where I've been involved, and they are both considered as high-risk organisms. One is on non-HIV infections, but are showing AIDS-like symptoms. So there has been several cases, and this happened like a few years ago, where patients, this was in Africa, in 3 countries in Africa, were coming to the clinic showing AIDS-like symptoms, but were HIV antibody negative with the routine tests that they use in Africa. So scientists were a little bit intrigued and bewildered, because they are showing symptoms of AIDS or of HIV infection, but were negative or seronegative for HIV. And then the other one is the approach that you can use even if your organism is or target is highly pathogenic, like the SARS-CoV-2. If you're developing a test or a diagnostic kit for that specific virus, you don't have to handle the infectious material if your kit is targeting nucleic acid. So those are the two case projects that I would present tomorrow, to just to give you a practical practical examples on how to handle novel, dangerous, or potentially infectious microorganisms. Oops. Sorry, it's hanging. Let me see. Oh, here, sorry. So in 1978, Pike and Salkin came out with an article giving a summary of the number of cases of LAIs in a span of like 48, almost 50 years. And during that time, there were over 4,000 LAIs, resulting in 168 deaths. And they were attributed to Brucella, Coxiella burnetii, hepatitis B virus, which I'm sure you're familiar with, Salmonella enterica, and this is a foodborne pathogen, Francisella tularensis, Mycobacterium tuberculosis, or the causative agent for TB, and Blastomyces, which is a fungus, and Venezuelan equine encephalitis virus, Chlamydia psittaci, and Coccidioides immitis, which is also a fungus. So clinical labs had 17% of these LAIs, while research laboratories accounted for 59%. And why do you think that's the case? I think in research labs, scientists tend to forget sometimes to observe good laboratory practice. In contrast, when you're working in a clinical lab, you're more conscious about pathogens, because you're very you're exposed basically. So you always bear in mind to wear the proper PPE and observe all the good laboratory practice like disinfecting your workbench and all those things. Sometimes in the research labs, especially if there is no systematic or there is no quality system that controls, activities, parang ano lang, parang kung minsan nakakalimutan yung good laboratory practice. So that's why they accounted for over 50% of LAIs. After the publication of Pike and Salkin's report in 2000, the number of LAIs reduced dramatically to just barely over 1,000. And the number of deaths also decreased significantly. And 5 of the deaths were of aborted fetuses as the consequence of a maternal LAI. So one example of a microorganism which can cause miscarriage or abortion would be listeria. So that's why if you're working in a food testing lab, they all here, at least in the States, they would ask you if you're pregnant, or do you intend to intend to become pregnant in a very short time. So they would, if you are going to work in the lab, they tend to assign you in an area or in testing where you are not involved on, or you're not exposed to listeria. And then the same causes, or the same causative organisms, almost the same causative organisms were reported also during that in that report. And clinical labs and research labs at this point were almost the same. So I think this gives some indication that research labs remain consistently not so observant of good laboratory practice or clinical lab practice, while the clinical labs have become a little bit more relaxed in their activities inside the lab. And those number of cases where they were able to identify the causative agent, there were some cases, or most of the cases of LAIs are unknown. So I think they could have been probably something that's new or not yet known at the time of their identification or research. And then the less obvious exposure, such as inhalation of infectious aerosols or direct contact of broken skin or surfaces contaminated by droplets, may possibly explain the incident responsible for a number of LAIs. And so this suggests that a lot of the LAIs could have been prevented if simple procedures such as hand washing or keeping your bench clean before and after work. So those little things, just like the health protocols, if we just follow them and observe them when we are working in a lab, those would help in reducing LAIs or contamination, basically. So I think those we have to always keep in mind that when we go into a lab, the mode changes, and you have to be aware that you can be exposed to a potential pathogen, especially if it's a microbiology lab. And the increase, they say that maybe the increase is really not because of increase in the number, but it's more of increase in surveillance. So just like for the SARS-CoV pandemic, if you test more, then you would discover more positives, basically. So they're saying that because of the increased surveillance programs, they were able to detect subclinical infections. And also, of course, it could be due to the use of inadequate containment procedures during the early stages of culture identification. So it could be because of the reduction in the number of incidents could be due to having improved containment procedures and also increased surveillance programs. And it is very useful for scientists to learn or know about occurrences of LAIs because as a research scientist, you will be able to learn lessons from LAIs. And there was, I think one report that they always cite is the occupational exposure with Brucella, which was actually an accidentally subculturing Brucella on the open bench. So it is very important to have the presence of mind when you're doing your research, especially when you're handling pathogens. And a small accident can cause or can result in a very, it can even result to a fatal fatality to your co-workers. And so it is always important to think of your co-workers and the environment. If not for yourself, you have to think of them. So the key message here is always be aware or always have a presence of mind when you are about to start your work in a microbiology lab. And I have already mentioned that because of publications of these cases or incidents, there have been improvements in containment equipment, engineering control. So they have the building of facilities have taken into consideration whatever LAIs were published. And then safety training has also become a requirement. For example, in our facility, we always have a minimum of 1 year or yearly refresher training, just because there are new hires or people tend to forget. So we always make it a point to have a yearly biosafety training. And this is because we handle also blood, because we have been working on HIV diagnostics. And so the premise is always think of the blood, even if it's safe. I mean, it's negative. It's it's tested to death, for example, where we get them from Stanford University Hospital or blood bank. And so it's tested with all kinds of viruses and all that. But still, you're never sure of whatever is present in the blood, which may not have been detected because there is still no test for it. So always treat it as potentially infectious material. So any human or animal body fluid or body parts, try to always remember or consider them as potentially infectious materials. And of course, there has been an evolution or national biosafety committees and guidelines where I think established in almost all countries throughout the world. And I noticed this especially around in the 80s. And it was also because of this genetically engineered plants, animals, and all that. So not just because of microbiology, but also because of other aspects in research. And they have this, when they call it a summary statement, agent summary statement, which describes the criteria to classify if your agent is a dangerous or a pathogenic microorganism. So if the agent is a proven hazard to laboratory personnel working with infectious materials, or it is suspected to have a high potential for causing LAIs, even though no documented cases exist, and or if the agent causes grave disease or presents a significant public health hazard. So it's what is the effect to the worker, to the co-workers, and to the environment would impact a classification of an agent. So these biosafety considerations or precautions are there to protect, again, the laboratory workers, the public, and the environment from exposure to potentially novel and infectious microorganisms that are handled and stored in the laboratory. So it is better to be more proactive rather than reactive to laboratory-acquired infections. So biosafety SOPs or precautions are there to prevent LAIs rather than to deal with it while it already happened. It's better to prevent it from happening. So what are the things or the risk criteria for establishing your containment levels? So if, for example, you're thinking of a project on isolating a disease-causing organism, you need to do a lot of research on that particular organism. For example, how infectious is your target organism? And then what is the origin of the agent? So whether it's indigenous or exotic, or does it have a reservoir, or is it transmitted from animals to humans, or zoonotic for that matter? So it is very important, or it is key to do a lot of research on your target organism. And importantly, what if it causes disease? What is the severity of that disease? Can it lead to death, for example? And how transmissible is your target organism? Is it highly transmissible like what we have now, like what SARS is showing? Does it have tissue preference or organ preference in the human body, for example? And then what are the hazards associated with the organism and the nature of the work being conducted? So if you're trying to develop a diagnostic kit for a new organism, it's very clear that you are just, you don't have to culture that organism. For example, if you're going to do a molecular test for SARS-CoV, and I will show you tomorrow how we did it without isolating the virus. And so, but if you are going to use it for example, for vaccine development, then that's a different story, because you may need to have tons of the virus. So you need to grow it in an environment where you should have all the safety precautions in order to prevent it from escaping out of the laboratory. So depending on what your goal or what your research is, then your facility requirements would also vary. So as I have said, get as much information on your target. Is it viral? Is it bacterial? Is it fungal? Is it a parasite? And do a risk analysis based on the potential hazard. So if you already have some, for example, SARS-CoV-2, it's a new organism, a new strain, but it has already 5 relatives that have been reported before, like MERS, MERS-CoV, or SARS. So you have an idea already about what is the nature of the virus. Is it enveloped, for example? Knowing all these physical properties of the virus would help you also develop a disinfection protocol or procedure in your laboratory. So, for example, if it's an enveloped virus, you know that they are very susceptible to alcohol or bleach. But if you have a naked virus, for example, like polio, they're very hard to, they're quite resistant. So you have to think of a different way of disinfecting your work area. And of course, you have to determine the capacity of your institution to handle PIMS, or potentially infectious microorganisms or samples, including the waste handling capability. So I know that we have very limited BSL-3s, for example, in the Philippines, and we don't even have a BSL-4, but that shouldn't discourage you from doing research on those levels, those types of pathogens. But at the same time, you have to also do something about having a facility or a mitigating measures to help or to prevent the organism from infecting you. For example, SARS-CoV was classified before as a level 3. So it's because it's highly transmissible, and they had to, in the beginning of the pandemic in the UK and here in the States, they had to be handled in a BSL-3 facility. But we had to think, we meaning the manufacturers of diagnostic kits, have to think of a practical way of handling the organism or the virus outside of a BSL-3. Otherwise, you can only run testing in facilities with BSL-3, which is not a very common thing to have. And so that gave us an idea also on how to handle the specimen. So there were several lysis buffers that were developed just to inactivate the virus, so you can work on a BSL-1, for example. So you don't have to, it doesn't even need a BSL-2 cabinet. So those are the things that you have to consider when you are going to do a research on, we call them the microbial discovery, or for our work, we call them virus discovery projects. And as I have mentioned earlier, I will give you also an example of in silico information from public databases, which would help you develop diagnostic kits, molecular diagnostic kits, without handling the organism if you don't have the capacity to start with. But of course, you can also collaborate with scientists who have access to this BSL-3 facility in the country. And the other way is to use surrogates, or the non-pathogenic relatives of your target. For example, if you're aiming for developing a test for listeria, you don't have to work on Listeria monocytogenes itself. You can start with the non-pathogenic relatives of Listeria monocytogenes, so that during the assay development, for example, you don't expose yourself or your coworkers to a highly pathogenic bacteria. Okay, and for viruses, you can always make armored RNA, or synthesize a pseudovirus and use them in your assay development. And what are the possible routes of infection or contamination that you have to think of? One is via contact, and you can be infected, or you can be contaminated just by decanting liquids, for example, or pipetting, or removal of screw caps. I'm sure if you're working in a microbiology lab, you have observed this, that sometimes when you decant something, you can have like minor, small droplets or small aerosols, which would eventually settle on your surfaces. And then vortex mixing of unsealed containers, which is a big no-no, okay? And then even streaking inocula on agar surfaces. If you're not very careful, you can be contaminating your bench area. And of course, when you're doing inoculation of animals. So contact route is a common route of entry. So you have to keep in mind, if you are working in a microbiology lab, I don't know, for me, I always do that. It's like, trying to disinfect the surface as often as you can, just to make sure that, whatever droplets have came off of your, during your manipulations, can be disinfected right away. And so also there are simple ways to avoid contamination. Of course, the use of gloves and other PPE. And you can also have an absorbent disposable covering on your workbench. We use that when we are handling, for example, blood samples or serum, plasma, or even swabs that we get from clinical sites. It's good to have that. It's like a diaper mat, and then you can just dispose of it as a potentially infectious clinical waste. And of course, it helps if you plan and organize your work. So, for example, if you're working in a BSL-3, you don't want to be going in and out because you forgot something outside. So you have to make a list of if it's helpful for you, and try to bring everything that you need into the P3 or BSL-3, and so you don't have to go back, go in and out, several times, because you have to wear the PPE and then remove it and then disinfect yourself. So it's a hassle. So plan and organize your work. And as I have said earlier, cleaning and disinfecting work surfaces is a very good practice. The other route is oral. So it can be through maybe a lot of you are young, from a younger generation. So when I was taking my microbiology course a long time ago, over 40 years ago, mouth pipetting was still being done in our lab in microbiology. And so it was not unheard of during those, during those years. But of course now it's a big no-no, right? And so you can use mechanical pipetting devices, and again, your practice of regular hand washing and not placing any objects, including fingers, into the mouth, of course, and wearing of a surgical mask or face shield. So this is depending on what you're handling, of course, right? So you have to have like a judgment call when you are what kind of PPE you are supposed to wear. And in some or most laboratories, they already have the set of PPE prescribed depending on what type of work or microorganism you are handling. And ocular route. So there's also a danger of viruses, for example, or bacteria going through your eyes. So wearing a face shield or safety glasses or goggles will protect the worker against splashing infectious material into the eyes. And inoculation route is if you're this usually occurs when you're handling a needle and a syringe for the transfer of materials from a rubber stoppered containers and for inoculation of animals. So when you are trying to raise antibodies, for example, against a certain virus in rabbits or in goats, you have to be very careful, because needle sticks can happen when the animals move. So when we were doing this animal injection or inoculation, it's always best to have another person holding on to or keeping the animal down to prevent it from moving around and you hurting yourself with the needle. And of course, when you are also injecting the animal, you can also create some aerosol of your material. So if you're injecting something that's inactivated, fine, that's it's safe, right? But if you're injecting something that's live and infectious, then you have to be really be very careful. And also animal bites and scratches. So this usually happens if you have animals or you're using animals in your research. And there is a rule that if you can avoid using needles and syringes in your research, try to avoid using it. So it's almost like your last resort. So of course, when you have to inject animals, you have to use needles, right? But there are some lab manipulations that you can substitute the use of needles by a different way. So try to avoid using it when you're handling infectious materials. And the other one is via respiratory route. And you can create aerosols when you're doing sonication and homogenization of your sample, centrifugation, and even just a vigorous discharge of fluids from pipettes or heating inoculation loops. So if you notice when you're just starting to heat your loop, there are some splashes or splatters of liquid. And so you have to bear in mind that you have to already prevent that, or maybe put some or just disinfect your surface right away. And of course, when you're opening lyophilized preparations and changing or during animal care in cages, so you can create aerosols if you are replacing, for example, litter or removing litter from animal cages. And as I have said, just simple manipulations of liquid suspensions may produce aerosols and droplets. And some of these droplets stay airborne for a while and can easily disperse throughout the laboratories, especially if you have this air circulating air conditioner, for example. And so, of course, if you are handling something infectious, you have to do it in a hood, right? In a BSL-2 hood, for example. But just in case, when you are, when this, in certain accidents, when you have these things escaping into the environment, then it gets circulated. And so it could also be transferred to other rooms sharing the ventilation of your ventilation system of your lab. And so larger particles tend to settle down. So again, try to disinfect as often as you can to make sure that you're protecting yourself and other workers in your lab. So there are 4 biosafety levels, which I think most of you know already. So you have the BSL-1, and this is just like handling of non-pathogenic strains of microorganisms which are not known to cause diseases in healthy adult humans. And examples, of course, is your very famous E. coli, or other than your other lab rats, as we call them. And BSL-2 consists of the moderate risk agents that cause human disease of varying severity by ingestion or through percutaneous or mucous membrane exposure. So you have, for example, the E. coli 0157, Salmonella, hepatitis B virus. HIV is actually considered a BSL-2. But when you start growing them and producing them in bigger quantities, then it gets elevated into a BSL-3. Okay, so in that case, you will be needing a different requirement or a different facility to be able to handle production of HIV in larger quantities. BSL-4, BSL-3 are those that are known to be highly transmissible via aerosols, and they can cause serious and potentially lethal infections. And usually they're indigenous or exotic in origin. And one best example is SARS-CoV-3. SARS-CoV-3 has a very unique way of getting into the human body and also into the different organs of the body, because it uses a receptor which is distributed all throughout our almost all throughout our organs. That's why if you have a critical case of COVID, you see it in your kidneys, in your lungs, in your heart. It's almost like overtaking the whole system of your body. So that's why it's very fatal. And BSL-4 are really the exotic agents that pose a high individual risk of life-threatening diseases, which are not, for which no treatment is available. And the famous example for this is Ebola virus. And so just to show you the requirements for each of this level, this is a CDC slide. All laboratories handling microorganisms should have this biohazard sign. And so the minimum requirements are, of course, controlled access. So if you have visitors, they should not be allowed to wander around without any staff accompanying them. And then a minimum requirement is a hand washing sink here on number 2, and a sharp hazards warning policy, and of course your PPE, which can be a pair of gloves and closed-toe shoes. So never wear open sandals or open-toed shoes in the lab, and goggles. This is more to protect splashes into your eyes. And of course, the bench. An autoclave is not a requirement, but it's something good to have. So you can, if you want to autoclave something like media, for example, so it's within the lab. Or you can use it also to decontaminate your waste. BSL-2 has all the BSL-1 features, but it requires a BSL-2 cabinet because this protects you and the environment from becoming contaminated in case you're handling pathogenic organisms. And in a BSL-2 lab, an autoclave is required. And so if you're handling, for example, blood samples, you can just use or handle them under the hood, like a BSL-2 hood. If you know already that the risk of having exotic or unknown viruses is not there, you can handle them here. But if you decide to grow your virus in larger quantity, like for example HIV, then you would be needing a BSL-3 facility. And the common feature or the requirement you would always see is that it has it has a double door, and it has a negative pressure inside. And this lab is fully sealed. So when you have to do a fumigation, nothing should leak out into the neighboring laboratories or neighboring environment. You are required to do fumigation every so often, just to make sure that the lab remains free of whatever you're handling with inside the P3. And so, of course, you have the hood. And then the green ones are not required, but nice to have, like a shower. So as soon as you come out of the P3, you can have a shower here. But the P3 that I handled in Cambridge did not have a shower, and because it's not required. And then you can have an air purifying respirator, which again is like an overkill, I think, if you're handling it. You handle it inside the hood, so that way you're protected from becoming infected. Sometimes it's very hard to have something in front of you like a shield, right? So sometimes it can even cause an accident. So if it's not needed, do not use it. And of course, this is a requirement. You should have an autoclave which goes one way. So if you have waste from here, it has to go through, clinical waste has to go through the autoclave and comes out to the other side of the P3 already autoclaved or sterilized. And also, if you have to bring in something that needs to be autoclaved, you can put it on the other side of the room, and then you can open it. Oh, sorry, this is one way, sorry. It's always just going out into it's meant for decontamination. And then these ones are just some things that are nice to have. So you have an exhaust HEPA filter or effluent decontamination system. And BSL-4, you need to have those nice to have in BSL-3, like the decontamination system and HEPA filters, supply and exhaust HEPA filters. And this is a requirement inside BSL-4, a positive pressure protective suit. So it's like, you see this in the movies where when they're handling dangerous viruses, for example. And so this is a must inside a BSL-4. And a shower is also a requirement as you go out of a BSL-4. And in BSL-3 and BSL-4, they usually have cameras inside. So if you're working alone and something happens to you, ideally you should always work in pairs. But in some cases where you're the only one working for that particular day, it's important to have those mirrors or cameras so that you can, if something happens to you, for example, if you faint, then somebody can see you and try to give you first aid. Okay, so what are the requirements if you're handling potentially infectious materials? immunization of lab personnel is required, at least where I work in the UK and here in the States. If you're handling blood samples, for example, they would ask you if you have received all the available immunization for bloodborne pathogens. So it's something that you should have, even if you're not handling that type of pathogen, just to be sure. And also they collect a baseline serum from you, and periodically they would also collect it just to monitor you whether you remain. For example, I was working on HIV. I had to they had to collect my blood every year and then run tests on HIV just to be sure that I did not get infected from handling HIV. And of course, good laboratory practices and training on proper transportation and shipment of specimens. So this is very important. There are rules to follow when you're handling or shipping pathogens or different categories of microorganisms. And of course, you should have a permanent and legible labels on your specimens. Again, this is, I have already talked about safe disposal and then training on proper procedures. And I forgot to mention that if you have a BSL-3 or BSL-4 facility, you should have all written SOPs, which have been reviewed by a safety officer. And it's important that the organization or the administration has accountability for the lab, especially if you're handling pathogenic microorganisms or infectious materials. And so waste management cannot be, I cannot overemphasize this because it's really very important to have the proper containment facility and disposal of infectious waste. I'm not so familiar in the Philippines, but I think these are all contracted out. And incineration is the best method of choice for infectious waste. Usually we contract this out to a third party who have been trained, and they come and collect our clinical waste or biohazard waste on a regular basis. But while you're keeping them in your lab, you have to also observe rules on how to package them, on how to seal the biohazard containers. So these are all written in your SOPs, and people working in the lab should be trained on them and given a refresher training every year. And final construction drawings should be available for future reference. So, and I have mentioned earlier that SOPs and plans should be reviewed by the safety officer before construction begins, and after the building has been completed, before work is allowed or work with infectious material begins. And of course, you should have a medical program just to make sure that if you're handling pathogenic organisms, you have to have a program. It doesn't mean that you have to have a doctor in-house, but there has to be a way or a contact person responsible for in case of emergencies, medical emergencies in the lab. And also, you have, as I have mentioned earlier, pregnant women, for example, are very vulnerable to Listeria monocytogenes or Brucella. So you have to review your employees' health or pregnancy status for female employees, just to make sure that you can, during the time of pregnancy, they can be moved somewhere else, or they can be deployed to another, for example, another department. And I have already mentioned this, that it's important to have an emergency plan, and also drills and simulated emergencies, like what happens if there's a leak or a spill in the lab, for example, of a highly infectious organism. And so there should be a drill to make sure that the employees understand what to do. And responsibility of the lab manager or director, there should be well-defined responsibilities, and of course the goal should be clear, and mandatory safety rules. And I have said already, written safety plans and a safety committee, and this should be effectively communicated to all the staff, and also emergency preparedness and auditing of laboratory operations. This is very important because you want to maintain the level of containment that you need to observe. So at least an annual full audit, and then you can have small audits in between, like quarterly or twice a year. So it's important to have an audit of your laboratory. And I have mentioned earlier that there should be a registry of the identity and location of infectious agents. This is very, very important because you want to make sure that no culture, for example, or no vial leaves the facility without your knowledge. So inventory is very important, especially because you can easily passage them, right? So the passage number has to be also monitored, so that it's really there's really a chain of traceability of your collection, because that's very important when you have to trace back where, for example, where a leak, for example, happened. And physical security, so it has to have limited access control and monitoring, and then personnel and visitor management, information security, and transport of biological agents. So this is in itself a separate seminar. And so I was just giving you an overview of cataloging and biosecurity. So as a final note, what is safety, or how do you achieve a safe workplace? And you can do this by really just following the good laboratory practice that we have been trained on as microbiologists. And of course, the local and national regulations, especially where you have to deal with transfer of materials from your laboratory to even within the same building, for example, there are rules that you have to follow, and especially if you have to ship it out of your facility. And lastly, common sense. So common sense plays a very key role when you are trying to decide, what PPE to wear. You do not over PPE yourself because it may even result in an accident. Okay, so that's it for today. And so tomorrow I'll be giving you 2 case studies or 2 case projects where I would show you how we handle novel organisms, and also if you're developing a test for SARS-CoV, you can develop a kit without handling the infectious material itself. So that's one way of avoiding it. Thank you so much for your time. I'm sorry I overshot again. And so if you have any questions, I'd be happy to answer them if I can.
Okay, thank you very much for that very informative presentation about Handling of Novel and Dangerous Pathogens, Dr. Nadala. I would just like to acknowledge the presence of our dear director, the DOST ITDI Director, Dr. Annabel V. Briones. Okay. Now, I'm sure that our participants are excited to ask their questions to our esteemed speaker. Let us now proceed to the Q&A portion. For our Zoom participants, please key in your questions in our chat box. While for our Facebook and YouTube participants, you may type in your questions in the comment section so that our moderators can pick them up and be answered by Dr. Nadala. Okay, so I think we have here our first question from Kim Mary via Zoom. Okay, for the first question, what would you suggest if we will isolate unknown viral pathogens from avian species? What biosafety level should be implemented?
So there have been a lot of publications or reports already that avian species harbor flu viruses or other viruses that could potentially infect humans. So since most of the respiratory viruses are transmitted through aerosol or the respiratory route, you should handle them at least in a BSL-2, but best in a BSL-3 facility. So again, depending on what your final objective is, the level of biosafety would depend on your end goal. So if you're going to just use it for not to grow the virus, but just to detect, for example, then you can put it right away in an inactivating solution or buffer. So then that way you can handle it in a lower level of biosafety, requiring a lower level of biosafety requirements.
Okay, thank you. For the next question, does having a biosafety officer a requirement as international standards for every laboratory in industrial research and hospital settings?
Yeah.
This is from Facebook.
Yeah, so biosafety officer is required wherever you are handling, whenever you're handling microorganisms or potentially biohazardous microorganisms or organisms for that matter. So, yes.
Okay, thank you. For the third question, this is from Lindley Suki via Zoom. Does having a clear mindset and good disposition a requirement for a researcher or scientist embarking on a microbial discovery? Is it also a safeguard if researchers also have a comprehensive psychologic and psychiatric assessment?
Let me divide this question into two. So the first part is, of course, having a good disposition is always a plus factor, especially if you're doing a discovery project, because failures can be your friend. As I will show you tomorrow, one of our projects was trying to determine the causative agent of a disease which was presenting AIDS-like symptoms, but HIV negative. So out of the 5,000 samples, we were only able to get one. So if you have a happy disposition or good disposition, that would help you overcome frustration or disappointments. And then the second part is comprehensive psychologic and psychiatric assessment. I think if you are working on a like a high security facility like a BSL-4, I think that would be a requirement just to make sure you are in the right frame of mind, because you can use this for bad purposes. So it has actually they do a background check, or it's not just psychiatric, but also if you had criminal records, for example, that's also a requirement for the high level security facilities.
Okay, thank you. And for our last question for this morning, for this afternoon, when working with environmental samples, what BSL level should be considered? This is from Facebook.
That's a very broad question. So it depends on what environment you're collecting from. For example, if it's just from water sources, you can have at least BSL-2 practices. Of course, you cannot bring that BSL-2, but when you're handling your samples, you have to follow the SOPs for handling samples of BSL-2 category organisms. But as I have mentioned earlier, if you're working with human body fluids, for example, or animals that can potentially have microorganisms or pathogens that can be transmitted to humans, handle your samples inside as if in BSL-2 at least, or BSL-3 if you have access to it.
Okay, thank you very much, Dr. Nadala, and to all our participants who actively engaged in our Q&A session. In the interest of time, we'll be ending the Q&A session now, but don't worry because we'll be raising the remaining questions tomorrow. So this concludes the first day of our two-day webinar. Please be reminded again that the evaluation form will be given tomorrow after the second day discussion. Answering the evaluation form is required before you can secure a copy of the certificate and a copy of the presentations as well. Okay, so thank you very much again, Dr. Nadala, and all our distinguished guests, and of course, all of our participants for today. Okay, see you tomorrow. Same Zoom link, same Facebook page, and same YouTube channel. This has been Red Danganan of DOST ITDI. Good afternoon, everyone, and happy lunch.
Thank you, Dr. Lu.
Thank you, Ma'am Annabelle.
Thank you.
See you tomorrow. Tomorrow,
Yeah,
practical
see you tomorrow.
example.
Okay.
Yeah.
Bye-bye.
Bye.
Ano yung 60,000?
Ay, go.
Kaibigan, huwag kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap ni Juan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Anumang hamon ay haharapin, mahirap man ay kakayanin.
Sa pinagsamang lakas at galing, tagumpay ay mararating.
Tara na, kaibigan,
kang magpaiwan. Gamitin ang dunong bansa'y susulong. Ating abutin ang pangarap-iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas. Ating abutin ang pangarap-iwan sa pamamagitan ng agham. Ang kaunlaran ay makakamtan kung lahat magtutulungan. Tara na, sama-sama itaguyod ang siyensya. Maayos na bukas para sa Pilipinas.
Ang pangarap ko ay maging piloto.
Pangarap kong magkaroon ng chief alternative for personal hygiene.
Pangarap kong magkaroon ng mabilis at murang transportasyon para sa lahat.
Pangarap kong masagot ang malnutrition.
Pangarap ko pong magkaroon ng effective communication means for emergency.
Pangarap kong ma-maximize yung renewable energy source and to reduce the carbon dioxide emission.
Pangarap ko pong maging scientist.
Ngayon na o simulan na, humanda sabay-sabay akyat. Hawak kamay tayo'y aangat lilipad.